Audience of The Ohio State University. Question, how many of you know someone who has or had cancer? Please raise your hands. Thought so. The sad fact is, a third of us in this room are going to get it. The good news is that 2/3 of us are going to do everything we can to help that 1/3. Cancer is a disease that basically makes its own enemies. When it takes one of us, it makes two of us more and more willing to fight back. So, what's happening in the world of cancer? Well, the problem is, it's getting worse. Not just in the US, but also in the developing countries, cancer's on the rise. And as the BBC put it, the cancer tidal wave is coming on. It's happened in February 2014. But there is another cancer that I want to talk about. It's especially deadly. It's ovarian cancer, and the reason is this. 24,000 women get it every year. 15,000 of them die, and most never have a chance. The reason is because ovarian cancer is diagnosed at stage 2, stage 3, by the time no therapy can actually make a difference. The reason for that is because ovarian cancer is so diverse that most single marker tests that you can do at the clinic right now cannot help you very much with diagnosing it. So why is that? Cancer is diverse. Well, to come to that problem, I actually got a speech a guy called Eric Lander gave at MIT, and he gave it for the Ig Nobel Awards, the funny Nobel Awards that you hear about, and he said something like this. He was talking about the Human Genome Project, which also is very diverse, and he says this, Human Genome Project, biology's moonshot, 15 years, 6 countries, $3 billion. 3 billion DNA letters, $1 a letter. Such a deal. 23 chromosomes, supposed to have 100,000 genes, turns out to only have 30,000, or maybe 25,000. Could be 40,000, check back next year. Should have the answer to everything, absolutely everything. Diabetes, asthma, cancer, evolution, life, death, taxes, even the Cleveland Browns. Only problem, there is no index. In short, Genome bought the book, hard to read. Eric Lander is a smart guy, very smart. And here's the point. The point being, back then, with so much information, we just couldn't process it. It was way too diverse, too much. And I'll give you an example of that right here. This is a complicated diagram, and no one's supposed to understand it. The point is, that's the immune system in one person. It has branches, branches has sub-branches, sub-branches has sub-branches, and it keeps on changing. Cells can change every second in your body. But the thing is this is the cure. Right here, somewhere in this diagram, is the cure for everything from peanut allergies to blood cancers. That's the problem. And it gets even worse because if you remember Mr. Lander, he said 25,000 genomes. Well, 25,000 genomes make more than 1 million proteins in your cell. You have 1 trillion cells in your body. If you can do the math, it's not pretty. But why am I talking about this? Well, my first love was much more regular, I would suppose. I loved coding. I coded my first program when I was 7. Not on that one. I use a much more modern computer, but that's the original computer they used to crack the German Enigma code. And then Gordon Moore came up with the wonderful integrated microcircuit, which Intel later pioneered. And recently, IBM came up with the first 3-dimensional microcircuit, which makes computing even faster and even more cheap and awesome. And science has been crucial, pioneering this approach for human consumption in terms of smartphones and tablets. But when I came into biotechnology with my love for all these microprocessors, I realized that in biology, we have a similar kind. It's called microfluidics. Most of you have never heard of it, but what it does is the same thing a processor does. You've got tiny millions of tiny wells, each of which is like a transistor, and tubing connecting it. Each of these wells can be used to process your DNA or make proteins for drugs. And the point is this, all of this can be done so much more cheaply because you're using so little of reagents. It's all really small. So when I looked into this and combined with my idea of my transistors, I looked for research and I found this interesting paper which my professor at Stanford, Steve Quake, published. Remember the immune system? Well, I'm just going to tell you how tools in microfluidics can make so much more sense out of that complete jumble. Right there is an example of a person whose antibodies were actually noted day 0 after he was immunized, day 7, and day 28. And the whole year later, the whole sequencing of his antibodies was done again. Year 1, day 0, year 2, day 0, and so on. The idea is the lines are correlating the antibodies. Look at this, though. This is the cool part. If you take a line and cross it between year 1 and year 2, you see a pattern. A pattern very commonly seen in biology. It's symmetrical. The immune system is doing the same thing it did 1 year ago, a whole year later, because it's the same vaccine. But this is even more awesome. There is one line that streaks across time from year 1 to year 2. It connects it, and that's something even more cool. That's an example of the immune system teaching itself from 1 year ago. Well, when I saw all of this, I really wanted to get involved. So I became a Pelotonia Research Fellow. I wrote a grant for a technology that I had an idea to make, and then we got the funding from OncoFilter from a national organization which accepted my peer-reviewed grant. And the result was the technology I invented, which we call the OncoFilter. We label it the living, breathing 3D biosensor, and the 3D comes from my original IBM microchip idea. And this is why it's cool, because for the first time, you have 5,300 times more area to process information, those little wells I was talking about. And you can do this 96 times faster. You can capture just 10 to the negative 12 grams per mil of protein on one chip. And you can do all of that with just 250 microliters of blood. And how does it work? Why does 3D matter? This is why. This is the ingenuity of the patent, really. You're looking at those spheres. Those are the wells. You have tubes connecting it. In 2 dimensions, you can only fit maybe 5 on a chip. But when you move those drainage areas down below, you can suddenly fit 3 times more. In fact, if you keep on expanding this, it's n times n. For as many rows, you have as many columns of capture areas. And this is proving instrumental in ovarian cancer, where getting multiple markers simultaneously is so important. Every chip is personalized for one patient. Every patient's proteome marker profile is captured, and that can tell you much better if that person has or does not have ovarian cancer. But how does it actually work? Well, that's the really cool part. That bead you're seeing right there, that has antibodies on it, and it's on a membrane that only lets fluid going through. The bead cannot, because it's like a mesh. The fluid is pulled through, and it basically has to go through the bead, because everywhere else is blocked, which means that the proteins bind to the antibodies much, much more than they otherwise would. And this allows us to detect the proteins using a kind of red marker, which basically the intensity of the color correlates to how much protein you have. So let's see. This is the channel. All those little dots everywhere are the membrane, and you can see the bead right there in the bottom. Fluid goes down the channel and down the bottom channel through the bead. So that's the bead, and as promised, that's the bead glowing with a protein called CA125, which is a fundamental protein for detecting ovarian cancer. But that's one step. What's in the future? Well, this is what I'm really excited for. Those are cells on the same chip. Why is that important? Because proteins are like products. They're finished products. The cells have made them. Cells are like factories. They produce proteins. I want to leave you with this picture. Imagine 1 million of such beads spread across this chip. Each bead has one cell, 1 million cells from your body, producing vaccines for you, producing drugs for you on your personalized chip. Cheaper, much more safe because it's your own cells, and much more efficient. But this is something cool I get this idea, I get this conversation always from other people telling me, but why should I care? Why does it matter? Why do I care about the diversity? Why not worry about it? Let me leave you with this final perspective. Everyone in this picture is someone who has made a difference in my life at my time in Ohio State. Every single person could contribute that difference because they were different themselves. And that difference, that diversity, is the reason their contributions mattered. Maybe if we stopped looking at the diversity in our body more from a negative perspective and start capitalizing on its strength, maybe then we can have a real killer app for cancer. I am an inventor because I love cool science and I love bringing it to light, but I am also an inventor because I love making a difference in their lives. That is my story. Thank you for being here.